Thank you very much for the kind introduction and for inviting me to be here today with you all. This is the first time I've been to the Allen Institute, but I've known of your work for a very long time and respect it and value it, and thank you for it. A lot of the data that you have created is actually going to be in this talk, so thank you very much. Over the next couple of minutes, the topic that I wanted to spend some time with you on is perturbation and control of human brain network dynamics. And so this is going to be a talk at a relatively large scale of human neuroscience, but I'm hoping that even if the particular questions and inferences that we make are not directly relevant to your work, the methods and some of the computational approaches might be relevant. So I'll just leave it there for now. Okay, so I wanted to start by saying that the human brain displays this really incredibly rich network architecture. So this is an image from diffusion spectrum imaging data that's acquired from a human in an MRI machine. We are using a relatively high resolution version of diffusion imaging, so the scan itself takes about an hour to acquire. We can we measure 720 different diffusion directions, and then from that high resolution diffusion scan, we can infer where white matter pathways are that connect up different pieces of the cortex and subcortical structures. So this is real data from a human. It was imaged by James Bartolozzi, who was actually an undergraduate in the lab, who subsequently went on to Pixar, because he creates such wonderful visualizations. Within that network architecture that we observe, these structural links physically constrain the propagation of activity at this very, very large scale, and in a manner that is dependent on the link topology. Understanding human brain network architecture, therefore, could provide some clarity on the basic neurophysiology and inform neuromodulation therapy. So if we can understand how this network architecture constrains large-scale propagation of activity, that could help us to understand how to intervene. Now, to get a little bit started on some of the basics of this approach, I wanted to just go through some preliminaries. So what we're going to do is we're going to take that image that I showed you previously, and we're going to model it as a network. So here, we're going to have every region of the brain be modeled as a network node, and then every structural link will be modeled as a network edge. Each of these network nodes, I will say that we tend to coarse grain the system into about 200 to 1,000 different brain regions that are anatomically defined, often based on cytoarchitecture or based on large-scale sulcal and gyral boundaries. We take that information and we condense it down into what's called an adjacency matrix. So this is an n by n matrix, where n is the number of regions in our coarse-grained approximation, and every element in this matrix tells you the strength of connectivity between node I and node J. And one of our very basic first questions, and that of many people in the field, is, are there simple principles of network connectivity in neural systems? To answer that question, we actually went across several different species. And so this is the mesoscale connectome from the mouse. Here's the Drosophila connectome again at the mesoscale. Here's macaque connectivity from Henry Kennedy's group. Here is the human diffusion spectrum imaging data, either at low resolution or at high resolution, so two different coarse grainings of the system. And what I'm showing you here is the weight of the connection as a function of the distance. And what you can see across all of these species and across very different ways of measuring connectivity, some being non-invasive, some being highly invasive, we see a negative correlation between weight and distance, meaning that the strongest connections tend to be short and the weakest connections tend to be long. And that's consistent over species and over methods of measurement.
Is this cortex or this is whole brain?
This is whole brain. But following that, you might want to say, is there a simple network model that would predict this relationship consistently? And one of the very earliest suggestions was, well, perhaps the architecture can be well explained by what's called a small world model. And this was motivated by early work from Watson-Strogatz in 1998 that was published in Nature, and where they wrote down a simple model where they said, well, if we can study what's called a simple regular graph, which is shown here on the left-hand side, and we slowly rewire it with a particular probability. So here is rewiring probability of 0, staying at the regular graph, and then a rewiring probability of 1, moving to a purely random graph. Then in between, we observe this small world architecture, where we have strong local clustering of connectivity and a few long-distance connections across the graph. So in this case, many short paths and few long connections can provide an optimal balance between local information processing and global information processing, or that was the suggestion of the model, okay? And so we decided to actually test that model more recently in the same data across multiple species. And specifically, we noted that one prediction of this relatively simplistic model is that these long-distance connections are placed randomly. So notice that this is a probability of randomly rewiring, right? So these long-distance connections have been placed randomly. If we look in neural systems, are long-distance connections placed randomly? If I had a show of hands, I'm sure I know exactly what you would all say. And it is in line precisely with what we see in the actual data. So just to walk you through this, here's the same connectivity from mouse, Drosophila, macaque, and the 2 resolution of human. And what we did is we said, well, let's calculate the similarity between node I's connectivity profile and node J's connectivity profile. If it is greater than expected under the assumption that long-distance connections have been placed at random, then that disproves the small world hypothesis, or at least suggests that that model is much too simplistic for accounting for neural systems. So here is the data. Here's the mean co-similarity between node I's connectivity and node J's connectivity as a function of how long the connection is. So this is the 5% longest connections up through the 25% longest connections. And in color, you see the actual data from the animal, and in gray, you see the data from a randomized null model. And what I just want you to notice is that the colored bars are consistently greater than the gray bars in mouse, in Drosophila, in macaque, and in the 2 humans. So what this indicates is that these long-distance connections are not placed at random. They are placed in a way that shares variance. So they're not independent placements. So that breaks one of the assumptions of the small world model and suggests that we need something more sophisticated. Probably not a surprise to you, but it's nice to be able to see it consistently across these different measurement types and species. So I think that motivates the question, what could be an alternative rule for link placement? In other areas of science, there's been this suggestion that, well, perhaps neural systems and other information processing systems are structured in a relatively modular way. So there's a group of nodes that are strongly and densely interconnected to one another. They form a module, and then there are long-distance connections between modules. So this is what the graph would look like if you have modular organization, and this is what the adjacency matrix would look like if you had modular organization. The shorter connections are inside of the module blocks, and then the long-distance connections are these ones that are on the off-diagonal blocks connecting one module to another module. Now, the benefit of an architecture like this is that, again, you get that nice propensity for local information processing inside the modules, as well as the integration of that information across different modules as well via the long-distance connections. Computationally, if you're interested in identifying this kind of modular structure in your data, we tend to use community detection techniques that have been constructed specifically for network-like data. And if you're curious about the specific versions that we use, I'm happy to talk to you about that afterward. But I want to note that even that modular hypothesis, I think, is too simplistic, because I think it's missing a lot of other ways in which information can be processed in a neural system. So the modular structure is really nice for information integration and segregation, but I don't think that's all that the neural systems are doing. We might also want to consider whether there are bipartite motifs that would allow for information transmission. So a bipartite motif is where there aren't very strong connections internally within one of these sets, but there are very strong connections between 2 sets. And that allows for one set of units to shuttle information over to another set relatively quickly without a lot of internal processing. This is what the graph would look like if you had bipartite structure, and this is what the adjacency matrix would look like if you had bipartite structure. So you see lots of connectivity in these off-diagonal blocks. But that's also not all I think that neural systems can do. And a third important motif, I think, is what are called core periphery motifs. And they allow for information broadcast to the rest of the system, and also for a set of units to receive information from everywhere else. And in a graph, you can see that architecture here. So here is the core. It has strong connectivity internally. It can broadcast information to the rest of the system and also receive information from the rest of the system as well. And then in the adjacency matrix representation, the core periphery structure looks like this, where here is the core, strong connectivity internally, and then connectivity to the rest of the system. Whereas the rest of this is blank, and that indicates that there's not very strong connectivity inside these three modules, or much connectivity between them either. There really is a focus of the unit on this core. So if all of these three different motifs could offer something important for information processing in neural systems, then perhaps what we really want is a way of taking a connectome-like structure and identifying where are the modules. Are there any bipartite structures? Is there any core periphery structure? And separately extract those simultaneously from the data. To do this, what we use is what are called weighted stochastic block models. And you can fit these weighted stochastic block models to the original data to extract regular modules that we're used to trying to detect, core periphery structures, and bipartite structures. Now, I'm not going to go into the math of the stochastic block model, but I am going to give you an intuition, which is basically that it is searching for a way of taking the adjacency matrix and delineating where these white lines should be such that the probability of connectivity inside a block is homogeneous. So in other words, this block has relatively high probability of connections, this block has low, this block has middling, etc. So it identifies a way of placing where these white lines should be, where are the demarcations between blocks inside of the adjacency matrix that would homogenize the probability of connectivity inside a block. So it's completely agnostic to whether the dense blocks are on the diagonal or off the diagonal. It doesn't care at all. Okay. So I'm going to show you two pieces of data that suggest that this is a really nice way of looking at connectivity structure. So on the left-hand side, what I'm showing you is we've taken the mesoscale connectome for the mouse, again, from here. Thank you very much. It's really beautiful. And then we have applied the stochastic block model to it to identify where these white lines should be. And then here what I'm plotting on top of it is patterns of gene co-expression. And what I just want you to notice is that the pattern of gene co-expression indicated by colors here is very nicely demarcated by these white lines, which have been defined from underlying structure. A similar example is from human data. So here on the right-hand side, we've taken the human diffusion spectrum imaging data that I showed you at the very beginning, applied a stochastic block model to identify where these demarcations should be, so the white lines here. And then the color is actually the resting state functional connectivity in the human. So that is patterns of similar neural activity from bold fMRI plotted on top of that structure. And again, what you can see is that where these white lines are placed nicely demarcate where strong functional coupling exists and where strongly negative structural functional coupling exists. So to me, this suggests that taking a more sophisticated model like the stochastic block model that allows for different motifs supportive of information processing is very useful in explaining the connectivity across neural systems. And I think should be what we should be using in place of the simpler small world model or even the less simple modular model that have been suggested over the last few years. Okay, so that brings me to the next point that I wanted to get into, which is that given the fact that there is this very rich network architecture in the human brain that we can now map out and try to come up with generative models for, we can also ask the question of how that network architecture constrains the propagation of activity. Because if we want to use this to understand dynamics of a neural system and information in any way, we need to be able to say something about the propagation of activity. So we've been struggling with this question for many years now. and specifically the point that much of the work that we've done in the past, much of the work that we've done in the past is just to take this architecture and try to describe it, to characterize statistically what are the motifs that we observe, what generative models might explain it. Much of it is descriptive and statistical. But what we'd really like to do is to have an understanding of when you perturb this system, how does it respond? And I think part of that comes from the physicist in me who says that, I'm not going to really understand a system until I perturb it and accurately predict what will happen. So what we'd like to do is to have a theory for how change in the activity of one region affects activity in other regions. It's challenging because the kind of data that we have is mostly this kind of data, which is structural data that tells us which pieces of the brain are connected up with one another. But given that that has a rich architecture, when we perturb the activity of one region, it's possible for that activity to flow through in a very constrained way these structural pathways to enact changes downstream. Okay, so in order to address this question, we have been looking at some of the techniques that have been developed in the field of network control theory. And specifically what network control theory was built to do is to say, how would you inject energy into a system in order to move it into a new state, knowing the underlying structure of connections between units. There are many different, it's an entire field, but the similarities that are present across the approaches are that you have to have an accurate understanding of what the structural connections are. So I need to have this underlying adjacency matrix. And then secondly, I need to have some model for how dynamics flow on this system. The very simplest possible model that you could write down is this one, which is just a linear, discrete time and time invariant network model, which says that the activity of the brain at time t plus 1 is equal to the weighted adjacency matrix, and so this is the structural connections, times the state of the brain at time t, and then plus some control. So B is a matrix with ones along the diagonal where we are injecting energy, and then U sub kappa is the energy that's injected into kappa regions at time t. Now, a lot of the really beautiful analytical work that's been done in network control theory has used this linear model, mostly because linear systems are much easier to deal with. They're more tractable to analytics than nonlinear systems. However, there are some really nice recent extensions of these approaches to nonlinear control, and although I'm not going to talk about them today, I think they have a lot of promise in the area of understanding neural systems as well. But since we're going to stick with the linear approach, what this constrains us to is talking about perturbations to the system in the vicinity of an operating point. So we're basically doing a linearization around the dynamical point where the system currently sits. Okay, one of the very first questions that we often ask when we are using ideas from network control to study a system is the system theoretically controllable? And given the structural connectivity matrix that I just showed you, and given the simple linear model of dynamics that I just showed you, you can write down what's called the t-steps controllability gramian, which is given here, this W, which as you can see is just a function of the adjacency matrix, and then this B matrix, which as I said along the diagonal tells you which regions energy is allowed to be injected into. And specifically what you can do is that you can study the smallest eigenvalues of this controllability gramian. If that smallest eigenvalue is very small, it means that it's practically very difficult to control the system, meaning you would have to inject a lot of energy to push that system into any state that you wish it to go. In our particular case, for the coarse-grained models that we have been studying, this smallest eigenvalue is very tiny, which suggests to us that practically this system is extremely hard to control. However, that's some numerical experiments. Oh, you're not able to see at the very bottom. There were some references. There's a paper by Tommaso Manera and Fabio Pasqualetti in 2017 IEEE TAC, which shows that using analytical experiments, you can also identify that this system is controllable given the linear model, but it's still practically difficult to do. So I think beyond that, what's probably more interesting to me is to say, if you have a network architecture in front of you or a circuit are there particular kinds of control strategies that that system is optimally placed to enact? Are there certain types of processes that that architecture is very good for? And particularly for the human brain, I think there are two kinds of control strategies that are very relevant to what we think about large-scale human cognitive neuroscience. The first one is in the engineering literature is called average controllability. And this is intuitively the ability to steer the system into many easily reachable states. The second type of strategy is called modal controllability. And intuitively, this is the ability to steer the system into lots of distant states farther away on the energy landscape. And to give you a deeper intuition for those who are in the sort of modeling sphere, here is what we do. So here's a schematic. The actual energy landscape is not of this sort, but this just helps you with visualization, where modal controllability allows you to push the system into a distant state, and average controllability allows you to push actually to all states, but you'll often go to the ones that are nearby on the energy landscape. Here, to derive what the actual definitions are, we still have to write down that simple linear model that we had up front and the t-steps controllability Gramian. The average controllability is then given by the trace of the inverse of the Gramian, where k is just one region that we're perturbing at a time. So you can ask whether a single region has the capacity to enact average controllability well or not. And then the modal controllability you can actually derive from the eigenvalues and eigenvectors of the adjacency matrix. So let's let V sub j be the jth eigenvector of your adjacency matrix. It has a particular eigenvalue lambda j. Then if the element Vij is small, so that's the ith element of this vector Vj, that means that the jth mode of the system is very poorly controllable from node I. Now what we can do with that intuition is that we can actually go across all n modes of the system to ask whether node I is capable of perturbing all of them. And so that's this one here. So here's the sum over all n modes of the system. And this expression critically has the Vij here squared at the back. And that provides a measure of the controllability of all n modes from region I. Okay, so these are the two statistics that I think are very useful in understanding some processes that exist at the very large scale in humans. But before we go to humans, I want to just ask the question of whether these two controllability statistics are similar to or different from what we would observe in common graph models. So here on the right-hand side, I have human brain connectomes, again, structural connectivity data. And then here are several other common graph models. The one that we talked about earlier today is the Watt-Strogatz model, so that's here in the light blue. But I also have the Barabasi-Albert preferential attachment model, a random geometric model, a couple modular models, a regular lattice, which was the left-hand side of the Watt-Strogatz model, and then a weighted random graph. And here is the modal controllability and the average controllability. This axis here is boundary controllability, which I'm not actually going to talk about, which is why I've almost flattened that dimension just so that you can mostly see modal and average. And what I just want you to notice is that where the brain sits in this space is quite different than all of the other graphical models, which suggests that the brain's capacity for these two kinds of control are quite different than what we would expect in something more simple. But now probably the more important question is, how do these statistics play out inside of the human brain at a regional level? So what we did is that we looked at each region of the brain, and we let each region be the controller. And then we asked to what degree that region produces a high level of average controllability or of modal controllability. And I've separated out the regional results into two different systems that I think are particularly interesting and relevant. In blue, what I'm showing you are the controllability values for regions that are in the default mode system, and those are highlighted in dark blue on the brain here. And what you can see is here's the degree of controllability for either the average strategy or for the modal strategy. And you can see that there's a strong preference for those regions to have high values of average controllability. Now, intuitively, what does that mean? That means that those regions have the white matter structure coming out of those regions places them in a spot that would allow them to push the system into many of the states that the system may need to go on the energy landscape. And then on the right-hand side are regions that are in what are called the cognitive control circuit. So this is frontal cortex, some parietal cortex, some cingulate, and a few other areas. And here you have the degree of controllability, either average or modal. And here I just want to point out that actually both kinds of control strategies are present and non-zero in these regions, but there is a clear preference for modal controllability. And so that suggests that these regions are placed in the white matter structure in spots that would allow them to push the system into other less easily reachable states on the energy landscape if energy was injected into them. So why is this interesting? Well, it's interesting because for the left-hand side, for these default mode resting regions, these are areas of the brain that are very active for us as humans when we're sitting, resting, doing nothing. They're sort of thought of as the baseline function of the brain. These are regions that are active when you're at baseline. So I think it's interesting to ask, if you had wanted to create a brain, would you want to put it in a state at its baseline where the regions that are active are the ones that can push you into most of the places on the energy landscape that you need to go? Possibly. That seems like a relatively optimal way of constructing the dynamical system. Separately, for the cognitive control regions, what I think is interesting about them is that those are the regions that, based on their white matter, not based on any computation that's being performed inside of gray matter, but just based on the white matter, they are located in places on the network that would allow them to push the brain into many different states. And the ability to push the brain into different states is precisely what cognitive control is thought to be able to do. It allows us to inhibit inappropriate behavior, to switch between different tasks, to stop impulses. So it's interesting to ask whether these white matter signatures are explaining partially the function that these regions are able to perform for us as humans. Now, I think this is just preliminary evidence to suggest that there's something here, but we followed up that study with a few others that I think take it a little bit farther. So that was the first study, where different regions of the brain have more or less power to alter whole brain dynamics, either in terms of average control or in terms of modal control. The second study was where we said, well, particularly if we think about that second idea, cognitive control, that changes a lot over the human lifespan, and specifically as kids grow. So executive function is one of the latest cognitive functions to develop for kids. So it would be interesting to ask whether network control changes over the same time period in human development that cognitive control changes. To ask that question, we studied 882 youth from the Philadelphia neurodevelopmental cohort, and they are from between the ages of 8 years and 22 years old. And for each of their structural brain matrices, or connectomes, we estimated their modal controllability and the average controllability. Every data point here is a child or a youth in that cohort. And first of all, what I want you to see is that the two types of controllability track one another. They're positively correlated via this exponential function. So that means that brains that have an architecture that is supportive of average control are the same brains that have architecture supportive of modal control. And this is actually not something that we find in many of the graph models that I showed you earlier. But the second point I want you to take from this figure is the age effect. So the brighter yellow are the 22-year-olds and the dark blue are the 8-year-olds. And what we see over the entire group is that younger youth tend to have low values of both kinds of control, and the older youth tend to have high values of both types of control. And what that suggests is that over this developmental period where cognitive control and executive function is changing very drastically, we are also seeing increases in the changes in the underlying white matter that support increases in two different cognitive control strategies. So that's another piece of evidence in support of the fact that this network control process may be supportive of cognition. The last piece of evidence, most recently, that we have put out is a study where we demonstrate that individual differences in these control metrics can explain individual differences on the performance of cognitive control tasks, and specifically on measures of impulsivity. So a human that has more impulsivity than normal has a decrement in control from the white matter structure. So together, these results suggest that the theory is a useful marker of how the brain enacts control to change network function. But you might want to ask the question, well, if these brains are changing over development and their controllability is changing, that's a nice correlative finding, but it doesn't necessarily tell me what is it about a network architecture that supports control. And if I'm going to make inferences about other species, then maybe I need to know precisely what it is about a network architecture that supports controllability. So to answer that question, this paper, I'm sorry that all the references got cut off. This paper is out last year in Nature Physics, if you want to look it up. It's a theory paper, so if you're on the more modeling side, this one might be the one that you're really excited about, where we basically ask the question of, if you have a particular architecture, can we predict how much energy would be required for that architecture to change state? And it turns out that dealing with an entire network is analytically really hard. We can do numerical simulations just fine, but doing analytics is really difficult. So what we did is that we simplified the problem down to just look at the bipartite structure created from the driver nodes. So those are the regions where we can inject energy into them, and then the non-driver nodes. So these are the regions that whose activity we wish to change. And just to drive home what a massive simplification this is, I'm showing you here on the bottom a full Drosophila mesoscale connectome, and then here the simplified version that we are studying, which is just the connectivity from the driver nodes to the non-driver nodes. So that we're just focusing on this tiny piece of the matrix and ignoring everything else. But using that simplification, we can derive an expression for the energy of a particular state transition. And I just want you to focus on the fact that in the denominator here is the determinant of Q, where Q is A21, that's this matrix here, times A21 transpose. So the fact that this little piece is in the denominator here, and specifically that it's the determinant of Q that it's in the denominator, gives us a really nice physical intuition about what it is regarding a network architecture that supports low energetics of state transitions. And to give you an intuition for that, we can kind of go back to what the determinant really means. And basically, we can study specifically, the determinant is related to the parallelotope that's formed by the vectors of weight distributions on these edges. So here what I'm showing you is the entire network that I just described, the simplified toy model. And then here are 3 different versions of that network with different weights on the edges. The weight on the edge is shown to you visually by the thickness of the edge here. So here what you can see is that there's one strong connection that goes from D3 to the non-driver node 2. This is a really relatively easy thing to control because you can just inject energy in here and it goes directly there. But this one is a little bit, less. This one is still relatively easy to control because you can inject it into D1 and it goes straight to the blue. However, this is much more difficult to control, and the reason is that if you want to change this blue node, you either want to change this or this, but both of them will simultaneously change this one as well because of the connections down into here. So this sort of interdigitation of connections from the driver nodes to the non-driver nodes makes the control a lot harder. And that is illustrated in the difference in the volume of the parallelotopes here. So what this suggests is that dissimilarly distributed weights on the edges into the non-drivers decreases the energy that is required for control. And that gives us an intuition for what it is about structure that provides a constraint on energetics. Now in the paper, if you're curious, we actually do a comparison from Drosophila to mouse and then to the human, and we find increasingly favorable network topologies for control as we go up in this set of 3. I think that's an interesting observation. It is only 3 species, so I'm not going to make a very conclusive statement about evolution allowing more complex types of control on network topologies, but I do think it does motivate future work that goes across different species to ask the question about what kinds of topologies support control in each of those species. Okay, so that brings me to the very last bit of data that I wanted to show you, which is that now that we have talked a little bit about the rich network architecture that's present in the large-scale human brain and how that architecture might constrain the propagation of activity. And we've specifically focused on endogenous processes like cognitive control and default mode activity, right? But now we want to ask the question of whether those same sorts of principles might be useful for us to understand exogenous control, so the stimulation of brain activity in the context of clinical conditions that require it. So if we want this approach to be relevant for optimizing and tuning brain stimulation, then we need something a little bit more definite and precise than what I've shown you so far. The average and modal controllability are these 2 statistics I've described, which ask questions about pushing to local states or distant states, but they've been completely agnostic to where you started, what was the initial state, or where you end, what is the final state you're trying to go to. They say nothing about that, right? So what we need, though, if we want to be able to use this approach for clinical interventions is something that is sensitive to where the brain currently sits and where the brain needs to go. So what we've done more recently is to ask the question of, well, if you have the brain sitting at this initial state here, and it would naturally go over to this state, this final state, but we want to push it over to this final state. How would we do that? And where should we inject energy, and at what time points, at which nodes in the brain, in order to push us from here to here? And we formalize this as an optimization problem. So we want to minimize this function, which has 2 terms. The final term penalizes the energy. So remember, U sub kappa is the energy that's being injected into the system. We want to minimize the amount of energy that we are injecting, because if we want this to be relevant eventually for clinical translation, lower energy is better for biological tissue. We also have this term right here, which indicates the distance of the current state from the target state. And by minimizing that, we're encouraging the system to take a relatively straight path from its current state to the target state that we want it to go to. We don't necessarily want it to be moving drastically around in state space on its way. So this is the minimization problem that we stipulate and then we solve for. And we can ask questions now about the specific state that we start in and the specific state that we end in. Now, before I get to the application to brain stimulation, and I have a few slides on that, I wanted to just gain a little bit of an intuition for what intuitions this approach could offer us for just a healthy human brain and endogenous activity. And specifically, this is work from Rick Betzel in the lab. He's now a professor at Indiana, and he asked the question, well, if we start the brain in any random state in silico, and we ask, what is the target state that we could go to that would require the least amount of energy? So let's start in a whole ensemble of random states, and let's ask, is there a common state, target state, that would take very little energy to get to from all possible beginning states? And I just want you to focus specifically on this middle panel here, which on the y-axis is the probability of being assigned to the target state. So these are the probability in which these areas are the ones that are active in the target state. And then on the x-axis is the log of the strength of node I. And there's a positive correlation here. And what that means is that the regions that tend to be active in that optimal low energy target state are the ones that have high structural strength. So those are the regions that have a high column sum of the structural adjacency matrix. So what and those are actually shown here on the right-hand side, just color-coded by whether they are in what's called the so-called rich club, which are areas that have strong connectivity to the rest of the brain and also tend to be connected to one another. And so what we observe is that those regions that would be active if you started from any state and went to a low energy state, or went to a state that required low energy, are the ones that have strong structural links to the rest of the brain. So that's a really interesting, again, sort of intuition builder that suggests that the way that the system is constructed allows for these low energy target states that are, again, relevant for the default mode of brain function. Okay, but let's get to the stimulation. So let's ask the question of whether we can understand how stimulation affects the brain and whether and to what degree the network control principles are relevant for explaining those processes. So for this, we use electrocorticography collected from patients who have medically refractory epilepsy. And here I show you along the x-axis is time, and then the y-axis is nodes, or brain regions or sensors. Here is the baseline activity. There's a pre-stim period. Then there is grid stimulation. So these are grids, not stereos, which means that there's a set of electrodes that are placed on the surface of the brain. These are not well, I'll leave it at that. I don't know how technical we should get into right now. So here is stimulation that's applied to one sensor, and then here's a post-stim period, and then there's an inter-stim period. Oh, sorry, that was right into you. Apologize. In addition to that, we also have these same patients. We have their structural connectivity, so we know for them specifically which structural connections are going from one sensor to the cortex underneath another sensor. We also have them performing a free recall task, and in a separate study that was led by Mike Kahana's group, we have a way of saying that the electrode power, to what degree is that electrode power in that particular person associated with good versus bad memory in the free recall task. What we'd like to do is to put these two pieces of data together to say, well, here is the state of the brain pre-stimulation, and we can determine whether it was a good memory state or a bad memory state or anywhere in between. So it's a continuous measurement based on this classifier output. Then we have the stimulation, and we know which region was stimulated, and we also know what the structural connections are between all the rest of the pieces of cortex underneath the other sensors. And then here we have the post-stimulation state, and again, this is power. And we can determine whether it was associated with good memory or bad memory or somewhere in between. Again, a continuous measurement. And we're going to use these ideas from network control. Specifically, we have the pre-stimulation state. That's going to be our X naught. And then there are two kinds of studies that we can perform. One is where we can do open loop control. We can basically just say, here's your pre-stim state. Then some energy is injected into node kappa, and we know which node was stimulated, so we know which region in that connectivity matrix has the energy being injected into it. And then we can ask, what are the states that it goes through next, and does it get to a state that looks very similar to the post-stimulation state? So in other words, given our model, do we get somewhere that's close to what we actually observe after stimulation? And then secondly, we can perform what the optimal control simulation, which is that we start from the pre-stimulation state, and we ask if we inject energy into this system and push you towards a good memory state, how much energy was required and which regions were required for us to put that stimulation into. So I'm not going to get into a lot of the particulars, but I wanted to show you these two plots just to give you an idea of where this has gone. This paper just came out in Cell Reports a couple of weeks ago. So what we have is, particularly for that second bit where we enact the optimal control transition from the initial state, so the pattern of power pre-stim, and then to a good memory state, we find that the energy required depends on the distance in state space to be traversed. So in other words, if the if the brain started in a state that was very close to good memory, it required less energy to get it to the good memory state, whereas if the brain started in a state that was farther from the good memory state, it required more energy in our optimal control stimulation to actually get you to the good memory state. So this positive correlation is present for each of the different individuals. So here are the 16 different subjects, and then in black is the over the entire group. And then secondly, here is the log of the energy. And again, but in this case on the x-axis is the change in probability. So here is where we have the change being from very far away, so poor memory, to good memory. And if you have a more drastic change, you are going to require more energy in the simulation. So those are just two sort of pieces of information that we actually then put into a random forest algorithm to ask whether we could predict the amount of energy that was required for a transition. Inside of this random forest algorithm, we place the transition distance, so that is how far you started from the good memory state. We also put in a measurement of the network topology, and this was based on the determinant of the Q matrix that I told you about earlier. And then we also put in the actual stimulation target, so which was the electrode that was being stimulated. And using these three pieces of information, we could predict the energy required for that transition with 93.2% accuracy, which is suggesting that we're getting something relevant for understanding the impact of stimulation on brain dynamics. The final point is that it's possible that this optimal control trajectory analysis that I've just described to you, in some cases can boil down to that simple modal controllability statistic I told you about earlier. And this is just some data. This is actually from Kambadi et al., Network Neuroscience in 2018. And here is, he shows that if you study the modal controllability of the region that was actually stimulated, it is positively correlated with a change in the memory encoding probability. This is a positive relationship, meaning that regions that are strong modal controllers inside the structure, if you stimulate them, lead to a change in the state that pushes you closest to the good memory state. I will note that there are very small numbers of subjects in these studies, so data collection in ECOG patients is slow and time-consuming, and we thank the patients and families for being part of the study, but we do need to take this with a grain of salt and wait for more individuals to go through this same stimulation regimen to understand if this continues to hold. But I think it's some initial evidence that's exciting and suggests further study. Okay. With that, this is what we have done over the last couple minutes. We started with trying to understand whether we could state some simple generative models for network architecture. We mentioned the simple small world architecture and also noted some of the ways in which current neuroscience data is demonstrating that that model is too simple. We went through modular structures, and then we went through the stochastic block model where we ended, and I suggested that that's really a nice generative framework for us to be using, I think, in the future, that allows for a richer array of architectural motifs that are supportive of different information processing capacities. Then we moved into understanding how network architecture can constrain the propagation of activity, and that's where we discussed the ideas of network control theory and asked the question of whether they can help us to understand the endogenous workings of the brain, both in terms of its default mode activity, but also in terms of cognitive control. And then in this final section, we asked whether we can use those same principles of network control to understand the effects of stimulation in patients with medically refractory epilepsy. And we showed some initial evidence from two papers, Stizo et al. In Cell Reports this year, a couple weeks ago, and then Kambadi et al. In 2018 in Network Neuroscience that suggests that the approach provides some important explanatory power in understanding the effects of stimulation. With that, I'd like to acknowledge the people who did a lot of the work. Many of the people who began these studies have actually now left and have faculty positions in various places. Some of the initial network control work was done by Shigu, who is now off leading his own group, and some of the later work from the Penn Philadelphia neurodevelopmental cohort was led by Evelyn Tang. Some of the early work in stochastic block models and in the comparisons across species was led by Rick Bexel, who's at Indiana. Evelyn is at the Max Planck Institute now. And the faculty collaborators that were particularly important in this set of work were Ted Satterthwaite and Raquel Gurr, who allowed us access to the Philadelphia neurodevelopmental cohort dataset, which has been really wonderful. And then Fabio Pasqualetti, who's a control theorist at UC Riverside in the Department of Mechanical Engineering. We were actually postdocs together at UC Santa Barbara and have stayed friends since. And obviously I'd like to acknowledge all of the funding, and particularly from the Paul Allen Foundation that has supported this work. And with that, thank you very much. I'd love to take questions.
That was really beautiful, thanks. I'm interested, or I might have missed some of it, but the degree to which your the matrix, and the dynamical predictions is consonant with more large-scale activity of the brain, either in the absence of any input or after a flash of a visual stimulus or something. How much of the variance can be explained with dynamical models from connectivity?
Yes. There are two papers that talk about that a little bit, although we're currently working on extending them in a couple different ways. But the two that I should just mention, because they were the first to address those kinds of questions, were this paper from Galan and colleagues in 2008, and then another one from Christopher Honey and Olaf Sporns in 2009. This one is published in the Proceedings of the National Academy of Sciences. And if you look in the supplement, they use a simple linear model of the dynamics, and they ask to what degree the functional connectivity that's elicited by that linear model and the underlying structural connectivity are similar. And so there's some preliminary evidence from that data set that suggests that bold fMRI time series at rest are somewhat predicted by this model. But I would say that task data is a completely different beast. And I would also say that bold, in our hands, the degree to which the model fits or doesn't fit depends some degree on the TR, so the actual sampling frequency of the data. Obviously, if you go into electrocorticography data, the beautiful, rich dynamics are very nonlinear. And so what we're doing by using a linear model for them is basically to just study the long time trends of changes in power, rather than the actual fluctuations of the electrical signal itself. Yeah?
So I have two questions I didn't understand. So in the middle part, when you talk about mean modal controllability and average controllability, how that changes with age, does the underlying DTI method take account of the degree of myelination?
That's a good question. There are separate measurements of myelination, not directly from the diffusion imaging, that have been proposed recently, but that are not currently included in the model. I think they would be very relevant to include, but they're not currently in there.
Okay. And then the other question I had is in the last part, when you talk about energy, are you actually varying the power or the current through the stimulating electrode?
Yeah, so they vary, not me personally, but yes, they are varying the stimulation frequency and intensity. So the patients, there was a pretty extensive regimen. So every sensor was stimulated at a range of frequencies and at a range of intensities. So it was a very long process.
And then you can predict those energies with a very high degree of confidence.
We can predict, well, so there are different levels of confidence for for changes in the intensity versus the amplitude versus the frequency. It's a lot more complicated. Yes, we can predict it with some degree of accuracy, but it depends on the actual stimulation parameters. We're better in some regimes than in others. Yeah.
Two quick questions. One near the end, it was very interesting to see how well you can predict the capacity to move the network from a state which is present in a random state versus a good memory state. But you never stated explicitly, were the patients becoming much better at doing the task if you moved them into what is classified as a good state?
Great, yeah, that's a great question, no. And the reason is that the stimulation data that we have is separate from the task data. And so what we need to do is exactly what you described, which is to ask whether patients can come back or a new set of patients can come where they do a stimulation that's not the entire battery that I just described, but just a few, and then do the task intermixed in between. So it's a new experiment. Fewer stimulations, large task sessions in between the stimulation. That would allow us to test that question, which we could only ask indirectly with these data.
And the second question, which is much more general, is the question of applying linear systems to such an incredibly complicated model in terms of local circuits, which are highly nonlinear. What do you view as what is the upper limit as to what linear dynamical systems would be as a good predictor for these types of networks?
Yeah, that's a good question. I mean, I think all you can really say based on prior work okay, so backing up a teeny bit, linear models and linear control are often applied to nonlinear systems that one would think would require nonlinear control. However, the linear systems have actually gotten the field quite far in technological, robotic, mechanical sensor systems, mostly because in many cases, you're able to predict the short time horizon effects, and that's enough to push the system in the direction that it needs to go. Following that initial push, you can basically capitalize on endogenous activity to take you the rest of the way, for example. So where I think this is going to hopefully be quite relevant is in understanding that sort of initial perturbation that's going to push you into something that's more an endogenous process afterward. So when I think about brains, I think that, particularly human brains, I'm not hoping or not looking for a method that will allow me to completely override the system to push it into somewhere else. I think what I want to do is to have a small perturbation that then capitalizes on endogenous activity, because I actually think that's where most of the natural power is going to come from, and so we should use it. I don't know if that answers your question. It was a bit vague.
I have a question here.
Yeah.
Yeah, great talk. So you mentioned that the brain has different networks, of course, the cognitive control, the resting state network, and I want to know, where did you stimulate and how stimulation had different effect, possibly depending on the regions on the grid?
That's a great question. Most of the stimulation was in the medial temporal lobe, so we have very little power in saying anything about stim in different networks of the brain in the way that cognitive neuroscientists talk about it.
There were striped grids inside,
Inside the medial temporal lobe, yeah. I mean, I think the data set that hopefully will help us to address your question a little bit better is transcranial magnetic stimulation inside of the scanner. So we currently have a faculty member at Penn who does TMS inside the fMRI. And in that case, we can ping regions that are in very different circuits, and then we can ask how that affects the rest of the system. So that's currently a Brain Initiative grant that we just finished the first year of, but hopefully we'll have more to say in a couple years. Yeah. Oh, you have the mic. Okay.
I wonder if you could comment, please, on your ideas for combining smaller scale or local circuit connectivity information with the large scale structural information. In particular, if a given brain region projects to another, but then has very diverse projections in the target region, that's the type of thing that you might expect to give you a lot of controllability in the target region, according to the intuition you gave us. I'm sure you've thought about this for years. Tell us what you think about the path towards bringing those two scales of connectivity information.
Yeah. So one path is to collaborate with you guys. The second path is that I think we have a little bit of work to do in understanding multi-layer control, because I think that's really what we need to address your question, which is basically if I have the pattern of connectivity, sorry, I use my hands a lot. If we have a pattern of connectivity at the very large scale, that's great, that's what we already have. But then if we have a second set of information that tells me what are the fine scale projections from this region to another region, I would actually use that as a separate layer. And then I would ask the question of, if you stimulate here, maybe that stimulation is a large area, particularly if it's TMS, right? You can't make that very small, but it's going to flow down into a smaller scale, which is going to then project in a very particular way to this other region. So we're currently working, we have 2 grants actually that are focused on this multi-layer. One is from the Paul Allen Foundation to focus on this multi-layer control problem. And I think that that's really interesting, because that will help us to at least have the math ready for data or insights that you and others like you have about what those predictions should look like. Yeah?
So we spend about a third of our lifetime asleep, so I was curious if you looked at or researched
I wish I spent
available functional data in sleep states, in different stages of sleep, where you can apply some of these models.
We haven't yet, but I think that would be really exciting. Yeah. There is some really beautiful work that's not in sleep, but that's from different levels of anesthesia, and it's by Xineng Qing, who's at Washington University, St. Louis in the electrical engineering department. He's doing really beautiful work combining control theory, neuroimaging data, and different levels of anesthesia. I feel like it's not sleep, but it's in that direction.
There's a lot of data on memory consolidation of the selective pruning of neurons, etc. during sleep. So
Yeah.
both in the non-human models,
I agree.
how the stages change.
Yes. Yep. I completely agree. I think that would be really neat to do. Thanks.
Now it does.
Okay.
So I was wondering about the conclusion that you can make about the controllability theory, given that you're using the method such as DTI, which provides the non-directional connectivity graph. So, for
Yeah.
example, using mesoscopic connectivity in the mouse, it's possible to derive the idea of the direction of the connectivity.
Yeah.
So, do you plan to extend your methods to more directional graphs, and to what extent the
Yeah.
linear perturbation theory might be applied to them?
That's a great question. So the method itself can definitely deal with directed graphs. Has a little bit more trouble with sine, although we're working on that more. But if you just have directed graphs, like the mesoscale mouse connectome, for example, the method still works. And in fact, the Nature Physics paper that I mentioned, we looked at the directed versions of the graphs for the main paper or the supplement. I can't remember which. One is in one and one is in the other. And then we symmetrize the matrices and ask how different are our predictions. And it turns out that if there's a small amount of directionality, the estimates of the symmetrized version are pretty good. And if there's large amounts of directionality, they become less good. In graph models that we can create that have large amounts of directionality, then the inferences are very different. But for the neural systems that we've been studying, including that connectome data set, the inferences are not wildly off. But if you want quantifications for that statement, it's probably in the supplement.
One last question.
Hi. Do you think that deep learning and reinforcement learning methods could help, especially in nonlinear control, and kind of like both finding control strategies and also excluding control strategies?
Yeah, great question. Again, I'm kind of going to have to punt because we're working on a project that uses reservoir computing to ask about control of those kinds of systems, particularly as they learn. And so I think there is a lot there. There is a lot that we can observe using artificial neural networks and extending the control principles to them that I think will be relevant for understanding humans. But it's ongoing work, and I think a lot of people around the world are trying to bring some contributions to that space. But there's nothing definitive I can say right now. But it's a good question and a good area, really exciting area, I think. All right, thank you.